Chen, you are a second-time awardee of our Birch Darshan Scholarship, which has supported your work now for, obviously, 2 years. Both men are here tonight, and I think they would appreciate knowing, as would all of us, what the investigative support has meant to you.
So the Birch Darshan Scholarship has helped me tremendously in the last 2.5 years. It's both inside my lab and outside the laboratory. Inside the lab, it directly helped me to buy the reagents and equipment that I needed for my research. And this research is to figure out how the brain differs between males and females on a biological level.
I wrote a book which was very popular called Why Men Never Remember and Women Never Forget, in an effort to explain some of the differences in behavior of males and females. Now, you work on the lowly worm.
Yes.
Tell us why you chose the worm as your subject.
So the worm is a very interesting organism for a number of reasons. First of all, it's really small. That means its brain is really, really simple. But by saying simple, I don't say it's not sophisticated. In fact, the worm has a really complicated nervous system in a way that it does not have a lot of cells to compose the nervous system, but it's intact. So it's a whole systematic nervous system that we can use a lot of tools, including molecular tools, genetic tools, and cellular tools, to sort of understand on a biological level where certain genes are expressed on a single cell level and how these genes are regulated. And also importantly, all of these can be done in a live animal during the developmental course of this animal. So I think it's important on a lot of levels because we're doing all of this with preserving the biological context of this animal.
What are some of the factors that impact gene regulation in your C. elegans animal?
So there are actually a lot of factors. We, in fact, just published this paper earlier this year that said basically the development or sexual maturation is one of the factors that influence or impact sexual differentiation.
Is that hormonal in the worm?
That's not known to be hormonal in the worm, but I think a very interesting thing is that we know on a genetic level, meaning there's a certain group of genes that express in both females and males. But if we try to see how these genes are regulated on a genetic level, they're quite different.
So talk to us about that regulation. The science is called epigenetics?
The science, it can be called epigenetics. There are many names for that. And a lot of people just call it gene regulation on a different level. So what I found is that these genes, they exist in both sexes, but during sexual maturation, only the male animals start to express these genes. And that means a whole lot because we know that the female worm and male worm actually do have a lot of different behaviors. And a lot of these different behaviors do also show up during sexual maturation.
Talk about those differences.
So the worms are known to behave differently sex-specifically. And the behavior I am interested in is actually locomotion. So if you just look at the worms for a long time or use a video recorder to record how the worms move on a little petri dish for a long time, and you quantify that behavior, you can actually find there are a lot of movement differences between the males and the females.
Who's more active?
The males are more active.
Are they aggressive?
I have to say, if you look at them for a longer time, they're kind of aggressive.
Are they larger?
They're actually smaller. They're smaller than the females. Yes. And many people think the differences are directly related to the fact that the males need to go find a female to mate. So that's why they're more active.
Whereas, small and mobile.
Yes, exactly. Right. So I found that one of these genes that I'm working on that turn on or get expressed during sexual maturation only in the male, if you deplete this gene from the genome, the males suddenly just become not as active anymore. and that's a feminizing feature of the male. So this means this gene is really important for contributing to at least part of these sexually different behaviors.
Talk about this newfound ability that we have, relatively speaking, of actually changing the structure of our DNA.
Okay, so I think you're talking about CRISPR.
I am.
So CRISPR is a technology that was developed a couple years ago. And now I think in almost every animal model that people are using for studying biological questions, we have CRISPR developed or being developed. So,
Which means what?
which means we have the ability to edit the genome relatively precisely compared to earlier technologies, and we're able to see, first of all, where these genes are located or expressed, I'd say, and when they're located or expressed, and very importantly, what happens if you deplete these genes from these genome.
I want to beg you to remember that in the whole science of CRISPR as it has evolved, it's very unusual to have an investigator think about the impact of sex, biological sex, on that gene if it's removed or, in other cases, inserted. So I hope you're thinking about, when you use CRISPR, whether the impact on a female and a male is different. What do you think about that?
That's actually exactly what we're doing right now.
Good for you. Good use of our funding.
So I have, in fact, personally used CRISPR to find where these genes are expressed in both females and males. And by this technology, I essentially was able to find endogenously, so meaning while this animal is still growing and developing, these group of genes are expressed only in one sex, which is the male. At a certain developmental time point during their development. So the next step will be to deplete some of these genes that showed sex-specific expression earlier to see if you deplete these genes from these animals, what happens to the sexually different behaviors.
Do you think there's a way to augment their function in the female?
If they're expressed in the female originally, there's a larger possibility that there will be, but so far most of the genes have found to be sexually different expressed in the male and not in the female.
But the same gene is present in both male
Correct.
and females. That's a very interesting point, to be able to augment perhaps the regulation or activate the gene in a female, and then the effect on behavior might be more similar to that of a male. Do you think that's possible?
That is quite possible, and in fact, some of the classic studies do use this trick to, let's say, force expressing of these particular gene or protein in an animal that usually does not express these gene or protein. We'll see what happens. That's, in fact, one of the very interesting points that we could follow up.
That's great. I hope you do, because
Yes.
we're very interested in the impact of biological sex and the epigenetic impact of environment on gene expression. I was privileged to know Marshall Nirenberg, who was one of the scientists who defined the genome, and he said one of the most interesting things to him was his sense of union with all living things, because 99% of the genome is identical, as you know, in all life forms. But what makes the difference between a worm and you, for example?
The difference between a worm and me, I think, first of all, I can't really see a worm just by my eyes.
Right.
They're microscopic, so the worm is only 1 millimeter in length, a full adult worm.
How do you explain, though, the similarity between your genome and this tiny microscopic organism, which is a model for you?
I think the differences between not only between me and the worm, but me and many other organisms on this world, is that they do, there's a lot of diversity in the animal kingdom, and different animals do have different behaviors. But in size, in autonomy, how they look and how they behave, and that's part of the beauty of the entire animal kingdom being very different. But from a very let's say from a training as a molecular biologist and a cellular developmental biologist, I do look at a lot of genes that exist in all of the animals,
Yes.
and these are the conserved genes. For example, these genes that I work on now, they exist in pretty much all animal species,
You included.
me included. Yes.
So it's the regulation or the activity of these genes that are controlled and that make us all different. Is that correct to say?
I think the regulation of these genes is definitely part of why we are different, and it's also part of why some of the regulatory mechanisms could be in fact similar. That's the basis of why we can use a little worm to understand the biology or biological mechanisms behind all these sexually different behaviors, to hope that some of what we learn from these experimental studies can give us insight into what higher organisms, including ourselves, behave.
I would like you to talk a little bit about your future in the United States as an investigator. You know, we've spent a lot of interest and funding on your rather brilliant career. You're full of promise. Will you be able to stay in the United States?
So right now, I am on a work visa. It's a H-1B work visa. I work at Columbia University and under a principal investigator, Dr. Oliver Hobart, at the Biological Sciences Department.
And you can't leave Dr. Hobart's lab to go to another laboratory, is that not correct, on a work visa?
It's possible. It's possible, and the work visa needs to be transferred. Well, it needs to be transferred.
How hard is that?
It's a process. The application of the original work visa took half a year. So I know I was going to get it because in a university, it's kind of granted that you are going to get the work visa, but because of, you know, things take a long time to get approved, and it will take another couple of months at least to get it renewed. So every time when this issue shows up, you do need to be very cautious about, you know, the timing and to count the days, basically.
What would be your next steps in staying here permanently if you wished to do that?
That's definitely the top choice, to have my own laboratory and to carry out my research with a group of my own people.
How technically would you have to achieve that with visas and green cards, and what would you need to do that?
So I am, in fact, in the process of applying for a green card right now. And that's usually, I think, what most people do. They work on a work visa first. And then on this work visa, during this process, they apply for a green card. And it takes different time to get a green card depending on where you are from originally. I personally am from China, and there are a lot of Chinese individuals in this country trying to apply for green cards. So essentially what that means is I'll need to wait for a long time, a few years, to get the green card.
How do you see your career if all goes well?
I ideally will be having my own laboratory, and I like New York a lot, so hopefully that happens in New York. And I'll be having my own students and postdoctoral fellows to work together to keep answering this question about, you know, the biological differences between males and females.
Well, that's of great interest to me, so I do hope you stay and solve some of the issues that arise. What do you think is the impact of on the individual of an analysis of the genome with regard to brain function? Do you think that by defining the genome of an individual patient, you will be able to predict the behavioral aspects of that patient?
I think that definitely makes it more possible, but I personally think as a fundamental scientist, as compared to an applied scientist or a clinician, in a way that I see doctors and clinicians and translational researchers as these group of great people who work directly with the patients, with essentially people who already feel something's wrong with them. So you see a doctor when something's wrong with you. So I see myself as this other side of the coin, where I work in a laboratory on not directly patients, but animal models that are just living their lives normally. They're developing at their normal state. And I want to figure out what happens when this animal is just developing normally. I think this is a very important aspect because I like to use the analogy of owning a car. If you have a car and the car is broken, you take it to a mechanic to see what's going on, and the mechanic will hopefully fix it for you. But to know how to fix a car, you first need to know how the car functions when it works.
Exactly.
So I see myself as one of these many scientists in this world to try to figure out how the animal work during its normal development.
Here's a question for you. Have you tried to disturb the environment of your male and female worms? And does that have an effect on the neurology and the behavior of these animals? For example, reducing the ambient temperature so that they're not at room temperature, for example. I presume your studies are at room temperature. Have you tried any of those environmental stresses on these worms, and does that have an impact on their neurology?
That's a very interesting question. I personally have not yet, but that's definitely down the road, because there have been actually studies that says when you don't feed the worm as well. So that's an environmental stress.
Yes.
They don't have enough to
Yes.
eat. They do behave differently, and this difference can be sex-related. So I of course want to see if these genes that I work on are related or contributing to these behavioral differences when the worms are fed well or not fed well when they're starved.
The next question is, can those changes in response to starvation or poor nutrition be transmitted to the progeny of those worms?
That's a very interesting question that some of the other people in our laboratory are actually touching on. So there have been studies that says that behaviors or phenotypes in one generation can be transmitted transgenerationally.
I'm sure you're aware of the studies on the Dutch famine, which was a period of starvation in Holland during the war, and the grandparents who father sons and then progeny, really those progenies suffer for a susceptibility for holding on to food, you might say, during development, which is sex-specific. A grandparent who's male will transmit that transgenerationally to his grandson. And so that's fascinating, and your work would be very important on how do we erase the impact, say, of poor nutrition on genetic expression. So I think the name of the game is in gene regulation, which can be permanent and transferable in some instances.
Right.
What was the most exciting thing about your research to you as you look back on your brief but brilliant career? What was the most exciting moment for you?
I think for this specific project that I'm working on, the moment that I discovered one of these genes that express or turned on only in the male animals and is completely not expressing in the female animals, that's the most exciting moment that I saw.
What has been most discouraging about your research?
So I think the discouraging part so I have been in this field since I got trained as a PhD student. And I think as an earlier student in your early career, one of the things you have to get adapted to is the daily repeat experiments that you need to do at your bench. A lot of these experiments, I'd say most of these experiments actually do not go into your either publication, or they don't give you too many conclusions, as so to speak. But those experiments are important to tell you what you cannot do to finish.
Like building a brick wall,
exactly.
piece by piece.
So troubleshooting, I'd say, is 90% of a biologist's life, And it can be pretty discouraging in the beginning, but over years you find out those are actually really essential part of your life to figure out what exactly is not working and how do I improve either technique or improve my way of thinking, or am I answering the wrong question and what other aspects am I missing to finally sort all of this out. I think, yeah.
What was your life in China like intellectually at the university before you came to the United States?
Okay, so I came to the United States for my PhD degree, and before that I went to college in China. And in these 4 years of college, in addition to taking a lot of coursework, I did 2 years of research in a laboratory that was, in fact, an immunology laboratory, completely different from what I'm doing right now. But that experience exposed me to what life could look like as a bench scientist. I worked with a group of very talented graduate students and postdocs and the advisor himself. And that, I think, prepared me in a way better to get adapted to my life here as in the United States.
What interested you in the differences between males and females?
So I became interested in the differences between females and males from a very young age, I'd say. I had a lot of pets, for example. And, you know, females and male pets do behave differently. And I experimented on other animal models, for example, the frogs. I used to work in the frogs as a grad student. I used to work on mice as an undergrad researcher. And females and males of pretty much all animal species do behave differently. So I think the interest in trying to figure out how these animals could behave differently has been in me for a long time. But it was only when I came to join Columbia at Dr. Hobert's laboratory that I started to realize, oh, I actually have the power as a genetist to figure out the biological mechanisms behind these behavioral differences.
That is exciting.
Yeah, that is really exciting.
Do you know the old biblical myth of how Adam was created as opposed to his wife?
Yes.
Well, Adam was the direct, according to the Bible,
Right.
creation.
Right.
From a handful of dust, all of his unique characteristics and behavioral traits were formed directly by God. Eve, on the other hand, when Adam became lonely and asked for a companion, was formed out of Adam's rib, implying that she was a clone of Adam and therefore genetically absolutely identical. And yet, their behavioral traits, even then, were quite different. So I find the myth very interesting. It's on the Sistine Chapel at the Vatican, the creation of Adam. And Eve is very much in the background, waiting to be cloned from this magnificent new creature, which is a kind of allegory of how women have been, or females have been looked at over the generations, 25 generations of academic research until recently. So I'm very pleased that you're interested in the specific biology of the inherent differences between males and females.
Yeah.
Is there anything else you'd like to talk about?
So when I designed this project, I had broken apart this big question of what's the biological mechanism that makes up for the differences between males and female into 4 very specific sub-questions. One of the questions is, where are these genes expressed? These genes that are important for contributing to the differences between males and female. And the second question is, when are they expressed? The third and fourth questions are, what are they doing to contribute to the sexually dimorphic or sexually different behaviors? And how are they doing what they're doing? In the first 2.5 years using the scholarship, I was able to find, to answer the first 2 questions. When are they expressed and where are they expressed? Now it comes the really exciting time to figure out what exactly are they doing, because we know there are sexually different behaviors in these worms, and what are these genes contributing exactly to these behaviors? And even more importantly, how are they doing these functions? So how are these genes regulated? Are they regulating other genes, for example, or are they regulated by other genes? So essentially, the complete pathway of all these gene regulatory network that ultimately contribute to the sexually dimorphic or different behaviors. So I think I have benefited tremendously from the scholarship to answer the first two sub-questions of my grant proposal, and now it's time to really answer the third and
Mr.
fourth.
Birch and Mr. Darshan are very close friends, and they wanted to make this grant to you as a partnership. And I'm sure they will be delighted as they watch exactly what you've done. So thank you.
Thank you.